Welcome, everyone, to the Ethel Browne Harvey Postdoctoral Seminar Series. My name is Colijn, and I'm a postdoc at Washington University in St. Louis, and we'll be moderating today with Shyama Nandakumar, a postdoc at the University of Pittsburgh. So we're excited to highlight the work of our outstanding postdoc members. So today, Austin Seroka from the Fred Hutchinson Cancer Center and Heather Dingwall from the University of Pennsylvania will share their research. Each speaker will give a 20-minute talk followed by 10 minutes of Q&A. So please type your questions in the Zoom Q&A box. I am pleased to introduce our first speaker, Dr. Austin Seroka. He completed his doctorate in the lab of Chris Do at the University of Oregon, studying the role of temporal patterning in the determination of synaptic targeting in Drosophila. In 2022, he joined the lab of Cecilia Mowens at the Fred Hutchinson Cancer Center to study organ innervation by vagus motor neurons during development. He's been well funded during his postdoc with an American Heart Association postdoc fellowship and then an F32 from NICHD. And today he will talk about how transcription factor code specifies vagal motor innervation of organs during development. Welcome, Austin.
Thanks so much for the kind introduction. And thanks again to the organizers for this opportunity to speak here. This has been a great series and we appreciate your time. So really excited to talk today about some work I've been doing to essentially understand how a pretty unique set of cells make some challenging decisions during development. So my background is in motor neuron identity and specification. And when I joined the Mowens lab, I became really interested in the vagus nerve or cranial nerve 10, which is the main highway of information transfer between the brain and the organ systems. There are two main components to the vagus nerve. There's an ascending sensory component, which is sensing things like our heart rate, our breathing, and transmitting that information to the central brain where computations are made, and then a motor component where those are turned into commands to control organ function. And vagus is part of the parasympathetic nervous system, so it does rest and digest functions. So things like slowing your heart rate when it goes too high, modulating blood pressure, ensuring proper peristalsis and digestion, and also things like pancreatic secretions. And just quickly, the vagus nerve, particularly stimulation, has been of high interest as a therapeutic target for the last 10 years or so. Usually the way this works is a pacemaker-like device is implanted and an electrode cuff is wrapped around the nerve bundle. And this is an FDA-approved treatment for drug-resistant epilepsy and depression and helps with recovery from stroke. But this is kind of a blunt tool, and because we don't know much about the diversity of the fibers in the nerve and which neurons are projecting where, there can be a lot of kind of off-target side effects of these treatments. So I'll make the case throughout my talk today that understanding the development and establishment of connections of these cells could really help to refine therapeutic approaches. So we address this question using the zebrafish, which as a vertebrate organism has the cranial nerves as conserved structures. So we can visualize these nerves by expression of the transcription factor islet-1, and we can identify vagus in particular by its location and morphology. And just to orient you to a couple things, the vagus cell bodies are collected in the brainstem in a structure called the vagus nucleus. They then project their axons out, and these fasciculate together before they split up to innervate different targets, being the pharyngeal arches and also a branch that projects to the viscera and the organ systems. Previous work in the lab has taught us quite a bit about how the pharyngeal arches are innervated. So two things to highlight. We know that there's a topographic correspondence where cells that are more anterior in the nucleus project to more anterior targets, and cells that are more posterior go to more posterior targets. And we also know, as shown in this movie, that the innervation happens sequentially. The more anterior targets are innervated first, followed by more posterior targets, and so on. And previous work has also taught us quite a bit about the spatiotemporal regulation of how this sequential innervation happens. And long story short, this is dependent on a spatial gradient of retinoic acid. And as the retinoic acid gradually recedes across development, it disinhibits these two players, MET in the motor neurons and HGF in the muscle targets, and kind of sequentially disinhibits them and allows the axons to perform innervation. But when I joined the lab, really my central question is, how can you have two neurons sitting next to each other in the brainstem and one knows to innervate, for example, something in the esophagus, and its neighbor knows to go all the way to the intestine? Kind of just an interesting basic kind of identity and pathfinding choice for these neurons. And so I'm really focused on this organ innervation aspect. And this is actually one of the first images I took in the lab, trying to visualize these deep, hard to see processes as they distribute to the viscera. And of course, there's a series of questions, are the same players involved that guide innervation of these more anterior targets? What's the developmental time course? And so one of the first experiments I did is just to ask, out of this whole motor nucleus, which of these cells are actually responsible for organ innervation? And to do this, we use this photoconversion-based retrograde labeling strategy. So we can drive the photoconvertible protein chiata under the control of islet 1. And we can then expose axon processes of interest to UV light and convert that chiata from green to red. And the converted protein will then diffuse back up to the cell bodies and reveal which cells are actually providing those axons. And so when we do this at 3 days, we can see that it's really just this posterior quarter of the nucleus, about 100 cells bilaterally, that are responsible for all of this parasympathetic organ control. If we do the same experiment 4 days later in development at 7 DPF, we'll still label the same subset of cells. So the population hasn't expanded or rearranged, but you can appreciate how dynamic this process is, how many sub-branches have formed. These cells are trying really hard to explore their environment and to locate the correct synaptic targets with the organs. And because we're working in zebrafish, we don't just rely on static images. We can also do time-lapse recordings and kind of watch this process happen in real time as the axons navigate. Okay, so we know it's this posterior quarter of cells in the hindbrain nucleus that are responsible for organ innervation, and we'd really like to understand some developmental rules that could be kind of regulating this long-distance pathfinding process. And when we started thinking about this problem, there are really two models that came to mind. One I'll call randomized search and find. So in this model, you have this population of cells, and due to some kind of stochastic developmental event, some cells acquire a fate, for example, to project to the heart, and other cells might acquire a fate to project to the gut. But based on other motifs of neurodevelopment and what we know about the more anterior cells, we also hypothesize that there could be some kind of topographic organization where maybe cells that are more anterior in this visceral population project to more anterior targets in the body plan, and so on. And so we wanted to distinguish these two hypotheses. And so to do this, I returned to the retrograde labeling experiments, except this time at a much finer scale. So here I've photoconverted processes, for example, from an anterior target, the pharynx, and you can see that it's this subset of cells providing those axons. And we can do the same thing from a posterior target, this subset of axons innervating the posterior gut, and see that we label only 4 or 5 posterior cells in the nucleus in this case. And if we do an intermediate target like the esophagus, we highlight this kind of discrete column of cells in the middle of the motor nucleus. And we can do this not just from different AP positions, but from distinct organ targets. So here are 2 experiments revealing the motor innervation of the swim bladder versus the gallbladder. And we can kind of accumulate these, do many of these experiments, and show a representation like this. And just to walk you through this, you can imagine the shoulders of these curves as kind of the AP boundaries of this red photoconversion expression across the length of the nucleus. And what we see is that really our second model is supported, that there seems to be a topographic representation where your position within the hindbrain nucleus tightly corresponds to which organ you're going to innervate during development. And there's also kind of this larger distinction between the anterior half of the nucleus, which are directly making NMJs on muscles, and the posterior viscera innervating half of the nucleus, which are actually doing something very unique for a motor neuron and providing presynaptic input to enteric ganglia in the organs. And I'll touch on this more later. And just very quickly, this topography, once it's formed, is very stable. So if I do this photo conversion of two different targets and we look at the position of these cells at 5 days, and then I let the animals swim for a couple more days and look at them again at 7 days, you can appreciate that the position of these cells hasn't changed once the topography is formed. So there's not new cells migrating in or reorganization. The topography is highly stable. And it's also not just in one dimension. I've talked about this AP dimension so far, but we've also seen through these labeling experiments that more dorsal cells go to more dorsal muscle targets and more ventral cells go to more ventral muscle targets. So it could be a 2 or even a 3-dimensional problem of kind of having this map in the brainstem that corresponds to where these axons are going to go. So would really like to know some, discrete molecular mechanisms that could be helping to specify these identities and guide the neurons to the correct target. And when we thought about this, we envisioned that we're probably looking for some kind of environmental cue or morphogen or gradient that's going to set up these distinct identities like I showed through the retrograde labeling experiments. And so to look for this, a former postdoc, Adam, and I built a developmental single cell atlas of cranial motor neurons across 4 time points. And long story short, we were able to identify not just our vagal cells of interest, but because we were purifying islet-1 positive cranial motor neurons, we also found distinct gene expression signatures for many of the cranial motor nuclei. So we found trigeminal and facial and oculomotor. And just to show you kind of what this looks like, we've done a broad HCR screen of about 100 genes across 4 different time points, helped by a fantastic postback in the lab, Italia. And we found genes, for example, a little bit of a different view here, a dorsal view. But we found genes that are expressed in all of the cranial motor nuclei, and then genes, for example, that are only expressed in oculomotor or trigeminal or facial or vagus. And we've also shown that we have the power in the single cell data, not only to look for these gross differences between the different cranial motor nuclei, but to look at spatial differences within the vagus nucleus itself. So for example, here I'm showing you again this dorsal view, just a couple representative genes we've looked at. So we found adhesion molecules and transcription factors only expressed in anterior, posterior cells, and really honed in on a subset of them. Here are 2 of my favorites, LHX4, which is only expressed in the anterior muscle innervating cells, and TBX5B, which is only expressed in the posterior organ innervating cells. And we found additional evidence for this dorsal ventral axis as well. We identified a gene HOXC6B, kind of this non-canonical expression of a Hox factor that is only in the dorsal cells in the nucleus and not in the more ventral cells. And we have a new talented graduate student, Veronica, who's going to tackle this question as her project. What's the role of this kind of non-canonical Hox expression in determining dorsal ventral target selection? So ultimately, we have this gene expression and we want to kind of correspond this with these different backfill retrograde labeling groups that we identified. And here's an example of one result, kind of a example of a result that we would be looking for. So here the experiment is I performed retrograde labeling from as far posterior as I can go in the animal. In this case, the axons going all the way back into the distal intestine. And when we look in the hindbrain, we can see that it's only really 2 or 3 cells at the very posterior tip of the motor nucleus that are labeled by this labeling, which makes sense with our topographic map. And in the single cell data, we identified a gene TAC1, which is involved in tachykinin, substance P, kind of satiety signaling, that's expressed in just the very few small number of the most posterior cells in the motor nucleus. So kind of trying to look for correspondence between spatially restricted gene expression and these populations of different organ targeting groups that we've identified through the retrograde labeling. We also want to do this rigorously and across many animals. So I developed a MATLAB pipeline to register all of these different HCRs and to do kind of detailed quantitation of this gene expression. So here I'm showing the vagus nucleus on the x-axis, and we have quantified HCRs of anterior gene, again, LHX4, and a posterior gene, TBX5B, and then a gene that's kind of expressed throughout the whole motor nucleus, islet 2A. And because we're registering these images, we can actually look at this cumulatively across many ends and kind of get these nice gene expression maps to especially use downstream in terms of like mutant analysis and trying to perturb these identities to have a ground truth. And so with everything I've shown you, we've looked at a lot of genes at this point, and this is kind of a schematic of some of the transcription factors and adhesion molecules we've identified and their expression domains within vagus. And we still have more work to do, but we have enough of these factors that have unique spatially restricted expression to kind of come up with a combinatorial code for different positions across the motor nucleus that we want to be able to test. We're also using this for tool development. So here I made a enhancer trap allele of TBX5B, one of our posterior factors. And very nicely, it only labels the most posterior viscera innervating neurons. You don't see any of the projections to the anterior arches. And kind of a zoom in on these projections on the gut with the gut lumen outlined here in purple. And we also want to manipulate these. We're working on a mutant screen, but one result I'll share is if we overexpress this posterior factor TBX5B in more anterior cells, kind of ectopically, what we see is that these anterior cells come down and they try to innervate the anterior arches, but they're kind of deflected off into outer space. They're not allowed into these branches. But TBX5 overexpressing cells are allowed into more posterior branches to go towards the organs. And so this is an interesting case where we have a factor that instead of instructing posterior targeting seems to actually be preventing anterior targeting of these posterior cells. So kind of more like a negative regulatory system. Okay, so again, what kind of like overall logic could be setting up these different transcriptional states within the motor nucleus? I brought in earlier retinoic acid that we know from Adam Isabella's experiments and from others that manipulating retinoic acid could affect axon targeting, but we wanted to look at its role in the actual specification of these cell types. And so what I'm showing you here is an experiment where we started treating the animals with either retinoic acid or a retinoic acid inhibitor very early in development at 24 hours, so about a day before they ever start making axons. And what we see if we look at our anterior transcription factor LHX4 is that if you treat with retinoic acid, you can really restrict the expression of this anterior factor and diminish it. And if you inhibit retinoic acid signaling, all of the neurons turn on this anterior factor. And we actually get the reciprocal relationship with posterior factors. So our posterior transcription factor, TBX5B, if we treat with retinoic acid, the whole nucleus becomes posteriorized. If we inhibit retinoic acid, the expression of TBX5B is gone. And we can confirm this through doing RNA bulk seq of treated animals and look for these signatures. But what actually happens to the axons? So here, this is the final experiment, but this is a little bit different. So here we're treating from 24 to 72 hours and then following up much later at 6 days after washing off the drugs. And so for our control animals, you have nice innervation of the anterior arches, innervation of the viscera. When we treat early with retinoic acid, we get really poor reduced innervation of these anterior pharyngeal arch targets, and we get increased branching in the visceral cavity. And really strikingly, when we inhibit retinoic acid early, we get almost no innervation of the viscera, and all of these axons go crazy and try to go anterior to innervate targets in the head. And strikingly, if we do these same treatments just a little bit later, from 56 to 99 hours, we see that everything looks like control. No differences from the control. And so what this suggests is that there's this early developmental window where retinoic acid and maybe other players can interact to set up these transcription factor codes that could be driving these unique columns or organ innervating pools to choose the correct target.
1 minute, Austin.
Thanks. And so in conclusion, I've shown you that motor neurons are organized topographically, that we made this single cell atlas, and that we've shown this involvement of retinoic acid. So what comes next? Just a quick plug that we're really interested in what Vegas is actually doing in terms of organ control. So we formed a collaboration with Isabel Espinosa Medina at Genelia, where we're going to try to answer this in terms of a circuit function. So here I'm showing we've identified the cells innervating the pancreas, the pancreatic islet in particular, and we want to do a panel of optogenetic manipulations to activate these cells or silence them while recording things like insulin release and calcium transients in the pancreas to try to understand what parasympathetic input to organ function is actually doing at a more mechanistic level. And so with that, thanks for your attention, and thanks to the lab, and I'm happy to take any questions.
Thank you, Austin. That was a great talk. These are some really interesting cells. So I want to ask the first question. So have you considered doing like a targeted depletion of the motor neurons, like maybe the posterior motor neurons after they've already developed, to see if any of the other ones could compensate?
Yeah, that's a really interesting idea. So Adam had done some experiments like that with axon severing, where he would kind of laser ablate different branches. The kind of blessing and the curse of the zebrafish is that everything regenerates. So if you ablate the cells, they come back, and there's kind of a whole paradigm that his new lab is studying about the regeneration aspect, because they actually can grow back to the correct targets that they initially chose. So that's a really good idea, and we do some of that work, but yeah, fish regenerates.
True. All right, we do have several questions here. So Chris Wright asks, are the anterior posterior patterns shifted in Hox complex mutants or morphins or RXR morphins?
Yeah, that's a really good question. So the Hoxes we're working on mutants. Hox B5, which does have expression in kind of that posterior domain. We showed in a paper, if you overexpress it, it can alter axon targeting, but the actual mutant analysis of Hox5 and the Hox6 that I showed in this talk is a work in progress. In terms of the retinoic acid players, we have constructs that Adam had used to either sparsely overexpress a constitutively active retinoic acid receptor or a dominant negative retinoic acid receptor. And those manipulations kind of had the expected consequence on the axon targeting. So if you turn up retinoic acid signal, you get a more posteriorized sulfate, and they like to go more posterior. But in terms of this complex network of getting the axons to the exact right place and forming functional connections with the organs, that's one of my future directions. We haven't really characterized. We've seen that these cells can be coerced to generally go more posterior in a direction, but we haven't seen that that's sufficient to get them to set up functional connections at the organ targets. But thanks for that question. That's a great question.
Okay, another question now from Ben Brissett. What are your thoughts on TBX3A's expression in both anterior and posterior segments of the nucleus? And is there a middle factor that inhibits its expression elsewhere?
Yeah, so thanks, Ben. That's a really keen observation. So what you're seeing there is actually kind of an interesting spatial expression pattern. So if I could show it like this, the nucleus itself, if you look from a dorsal view, is kind of curved like this, almost like a C shape. And the TBX3A expression in the hindbrain is a straight stripe. So what you're actually seeing is that the posterior and anterior tips of vagus are within that TBX3A expression domain, and the middle is kind of curved more superficially and it's outside of that expression domain. So that's one we were kind of puzzled by, where we were thinking about kind of complex ways you might set that up, but it seems like actually is more about the kind of 3D shape of the structure and where different cells in the nucleus end up. But that's a great question.
So just a reminder for everyone, if you do have a question, type it into the Zoom Q&A box. But Ben has another question for you.
Sure.
So what is the source of the retinoic acid? Does its location suggest any contribution to the dorsal ventral axis that you described?
Yeah, so during development, the retinoic acid is coming from the paraxial mesoderm. So we know where it's coming from. What's interesting is that these retinoic acid dependent mechanisms that I talked about in terms of the anterior arch innervation and early setting up these fates, this retinoic acid gradient has fully receded by around 3 days, 3.5 days. And that's actually when the visceral motor axons are navigating and performing pathfinding to locate the organ targets. And so we really think anything retinoic acid is doing is probably this early kind of spatial patterning of the nucleus, in which case it could be involved in dorsal ventral patterning, even though the gradient is really just along the AP axis. But you could imagine ways you could have differential response elements to the retinoic acid gradient turned on or off in the more dorsal or ventral cells, maybe by something like BMP or that's a great question. But yeah, right now we don't really know the involvement between retinoic acid and the DV patterning.
Cool. Okay, I have another question for you. I'm a motor neuron amateur. So these cells must be like the longest cells in the whole. As the fish grows, do they break into several cells? Do they proliferate and somehow become more than just one really, really long one?
Yeah, that's a great question because especially like when I introduce this, thinking about the human, that you have neurons that are coming from your brainstem and going all the way to the intestine and the colon is really remarkable for a single cell to do that. Um, from what we understand, they just grow with the fish and they're plastic enough to just adapt. But something I'd be interested in doing eventually is looking at later stages. I've really only looked at these animals up to the latest 14 days. And the fish are still pretty small at that point. So what these structures look like in adult fish that are 2, 3 times the size, I think that would be really interesting. Yeah.
Cool. Okay. I think maybe Gage has a question. Go ahead and please, Gage, type your question.
Oh, yeah. Oh, you want me to type it in? Because I wasn't able to type it in the Q&A, maybe because I'm a panelist. Can I just ask?
Let me see if I can do it for you, speed round.
Or I can type it. Yeah, I wasn't able to.
Maybe just go ahead and ask it since you're already talking.
Okay. Yeah, no, I was just wondering, yeah, really beautiful talk. I really enjoyed that. I was wondering if you could, I know like Tom Schilling has sorted out like CADA converted cells for RNA sequencing. So I wonder if you've thought about sorting out the different nuclei from the retrograde tracing to really start to look more comprehensively at gene expression differences between the different nuclei and how that might correlate.
Yeah, that's a great suggestion. So Adam Isabella did try that. He took cohorts of fish and would photo convert. He really worked on the arches, so he'd do like PA3 or PA4, PA5. We just ended up with a signal to noise challenge that it's so few cells per animal that even with really strict sorting, we just had a hard time getting good signal to noise with bulk seq.
Okay.
But I'd like to think of other maybe, cassette-based lineage recording with single cell, some other ways to try to get at that. But that's a great idea.
Yeah.
Okay, thank you so much, Austin, for a great talk. Thank you.
Hey, thank you, Sarah. And thanks, Austin, for a great talk. I'm Shyama, and it's my pleasure to introduce today's second speaker, Dr. Heather Dingwall, who's a postdoctoral fellow in the lab of Jana Kambarov at the University of Pennsylvania Perelman School of Medicine. Heather earned her PhD in human evolutionary biology from Harvard University, where she was co-advised by Dr. Terence Capellini and Dr. Jenna Galloway. Heather's research is uniquely situated at the intersection of developmental biology, functional genetics, and morphological evolution. As a PhD candidate, her work focused on the developmental genetics and evolution of tendon growth, a critical area for understanding musculoskeletal biology. And her current cutting-edge work that she'll be discussing today will focus on dissecting the molecular mechanisms underlying the formation and evolution of ectodermal appendages, including sweat glands. This work has already led to some high-impact publications, such as in Developmental Cell. And beyond her impressive publication record, Heather has been extremely well-funded throughout her career, including having secured the NSF GRFP and NIH 332 postdoctoral training fellowship, in addition to grants from the Dermatology Foundation and a pilot grant at Penn. Heather says and has shown her deep commitment to mentorship, service, and public outreach. And Heather is also on the faculty job market, so I'm sure we'll see a lot of exciting research emerge from the future Dingwall lab. Please join me in giving a very warm welcome to Heather. Please take it away.
Okay, thank you so much for that introduction. I am really thrilled to be here and be able to talk a little bit about some work that we're currently in the process of putting together for publication. And hopefully from this picture of now the late, great Jane Goodall, you can see just visually that there's at least one key visual difference in skin between humans and our closest living relatives, the chimpanzee. This chimpanzee here is named David Graybeard, and you can see that he has a thick fur coat and Jane does not. In addition to lacking fur, humans also are covered in eccrine sweat glands across our body skin, which is unique to humans. And together, these two features, having no fur and a lot of eccrine glands, enables us to thermoregulate via evaporative cooling. And that works by having an eccrine gland whose coil resides deep in the lower layer of the skin called the dermis, and pumps water and a little bit of salt up through the duct, which gets deposited on the surface of the epidermis. And then that will remove heat from the body and transfer it into the air as it vaporizes off the skin, thus cooling us down. And an inability to sweat is completely incompatible with human life. If you cannot sweat, you will die. And so this is sort of thought of as being a really important feature in humans during human evolution, as well as for modern human health. And when my postdoc advisor, Jana, was a postdoc herself, she showed that not only are humans completely covered in eccrine glands, we actually have an eccrine density that's about 10 times higher than our closest relatives, the chimps, and our distant relative, the macaques. But macaques and chimps are able to grow eccrine glands out in their body skin. It's just a very, very few of them. And although we may look furless, we actually do not differ in our hair density at any body site in our body skin. from a chimpanzee. And so actually, even though it looks like we don't have any hair, we're actually covered in these tiny, short, thin, unpigmented hairs called vellus hair. And so that sort of was the state of things when I joined Yana's lab as a postdoc, was just this observation that human vellus hair is unique, and humans are uniquely covered in eccrine glands, and that sort of this ability to build some eccrine glands in body skin is a unique primate innovation in evolution, because most mammals that can make eccrine glands anywhere will only make them in their paws, and they're actually used for grip, not for thermoregulation. And so with the help of a really amazing high school student who returned as an undergrad this past summer, and a collaborator at the University of Buffalo, Joyce Siriani, who herself did her PhD on comparative primate development in the 70s. We wanted to know when macaques form their eccrine glands during development, because it was previously known already that in humans, although we have a lot of eccrine glands and hair follicles in our body skin, we actually don't make them at the same time. So the human skin makes a bunch of hair first, and then we'll form eccrine glands later. And so in the macaque, even though they don't make a lot of eccrine glands, what Felice did is she looked through a lot of sections that we H&E stained from these beautiful macaque, timed pregnant macaque specimens from Joyce. And she actually identified that macaques, like humans, also form their eccrine glands later than hair, in sort of a space that's already populated with mature hair, suggesting that this offset, this developmental offset between the two appendage types, actually dates all the way back to our common ancestor with catarrhines, which includes old world monkeys like a macaque, African apes, and humans. And so based on this, we were really curious about what, if any, rules there are for the relationships for how eccrine glands are actually sort of populating this space that is already occupied by hair follicles. So taking all of those histological sections of developing human and developing macaque skin, we just took a bunch of measurements to try to understand differences in the size of the appendage. So on the left, I'm showing you how much space an eccrine gland takes up in a sagittal section, how much space a hair follicle takes up. And we initially expected the macaque hairs to be much bigger than the human hairs already during development, because they are much bigger later in adult life, but actually they're only marginally bigger, so that was already surprising to us. And we also found that hair follicles and eccrine glands sort of are spaced apart more than we would have expected, especially in the macaque. So the macaque seems to not be able to make eccrine glands very close to hair follicles at all, whereas in humans, they can make them almost as close as you can make an eccrine gland, but not quite. And so to try to go beyond just this one-dimensional measurement, really, in sections, we decided to try to understand how these relationships combine and scale to a 2D skin region. And for that, we had to collaborate with a mathematician, George Kambarov, who happens to be Yana's father, from the University of Alaska. And so he identified this as a pretty classic circle packing problem or sphere packing problem. And so what he helped us do is, using the empirically determined measurements, he then was able to put them together to create this model of how in a two-dimensional plane, this is as if we're looking down on the skin now, not into the skin, you can fit appendages of different types and different combinations into a space. So this is just sort of a dummy model showing the appendage in this darker gray circle and this larger, what we're calling an exclusion field in the larger circle. And then the model is just trying to pack as many possible appendages in there as it can. And so if we look at the actual data that went into this for different species and different appendage pairs, I think you can already see that the macaque exclusion zone around a hair relative to an eccrine gland is much bigger than it is for a human, and than it is for eccrine to eccrine. I will note that in this, all of the data that I will show you, we're using the human eccrine parameters for the macaque, as we did not observe any adjacent eccrine glands in the macaque, although we're in the process of running different versions of this model to try to push it to the extremes to see what's possible. So the ground state for what this looks like is on the top, you see blue circles in a plane. Those represent hair follicles forming they're the only appendage that's allowed to form in the plane. And on the bottom are eccrine glands, and this is what it would look like at maximal packing. And so the top state is sort of what happens first during development, and then what George then did was try to get the model to then make as many eccrine glands as possible within this space that's already populated by hair follicles. And we're assuming a maximal packing scenario, sort of just to test the hypothesis that humans have attempted to maximally pack their appendages. And the model is also assuming that these two programs are occurring separately, and then the fact that the hair follicle is there is sort of inhibiting something about eccrine development. So when you try to maximally pack eccrine glands in a hair follicle plane, this is what it looks like. So on the left, you can see in the human, the red circles are eccrine glands, and they're sort of interspersed throughout. And this sort of gives us a roughly a 1-to-1 ratio of hair follicles to eccrine glands in this plane. In the macaque, though, you won't see any red eccrine glands, and that is because the model predicts that the macaque should not be able to make eccrine glands at all. And so for the human, this recapitulates what we know to be the observed ratio of hair follicles to eccrine glands in the human face. In macaques in the face, the observed ratio is about 17-to-1. So very, very, very few eccrine glands, but still more than 0. And so in subsequent iterations of the model, which I'm not going to show you, George sort of altered some parameters to see what would have to happen to get the macaque to even make an eccrine gland. And it turns out that you have to fail to form about 10% of the hair follicles before you can even start to make an eccrine gland. And so what this has sort of told us is that the spacing of the hair follicles initially, earlier in development, gives human skin the capacity to build eccrine glands in there in the first place. And so there's just something different about how human hair and macaque hair is sort of setting the plane and the fields in which eccrine glands can form. But in order to actively make an eccrine gland, you need something else. And that thing is the transcription factor engrailed-1. So engrailed is required for eccrine gland development. If you conditionally knock it out in the basal epidermis, you will not form any eccrine glands. And also, previous work from a former postdoc in our lab, Daniel Aldea, showed that humans evolved a cis-regulatory mechanism to express very high levels of engrailed-1 in developing skin. So he identified this enhancer that he called ECE18 that accumulated 10 human-specific mutations that enhance the activity of engrailed-1. He showed this both in vitro and in vivo, and in the humanized mouse version of this enhancer, the mice actually make more sweat glands in the soles of their feet. So knowing that engrailed is required and that humans have evolved this mechanism, we then decided to look at when and where is engrailed expressed in developing human skin. So on the right, on the left, you can see a section through a 108 days post-conception fetal chest skin. And at this point, no hair follicles have formed, or no eccrine glands have formed yet. Right here, you can see a hair peg. That's sort of the second stage of hair development. And you can see a nice hair placode over here. And in yellow, you can see engrailed is kind of just very minorly sprinkled throughout the basal epidermis. By 132 days post-conception, you can see this thickening in the basal epidermis here that has a lot of engrailed in it. That's an eccrine placode. And over on the right here, you can see a nice big hair follicle that has already formed. And you can see that engrailed is, again, sort of sprinkled throughout the inter-appendage epidermis, as well as a bit in the mesenchyme. And then by 163 days post-conception, you can see this beautiful nascent stage eccrine gland. So at this point, it has grown down into the dermis. And you can see this expanded portion here. That's where the cells that will become the secretory portion of the eccrine gland are actually going to start differentiating if they have not already started already. And you can see that those are very high for engrailed in the nascent eccrine gland. And then up here, you can see a little duct of a nascent eccrine gland as well that are very high for engrailed. And again, it's still kind of in the interfollicular epidermis. But I would like to draw your attention to the fact that over here, you can see these beautiful hair follicles that are also positive for engrailed in multiple populations within the hair follicle itself. So it's in this population called the outer root sheath, which is a basal population, but it is, it is also sort of deep in the bulb here. And that's important because, so like I said earlier, human hair actually doesn't start out that much smaller than the fetal macaque hair. And it actually, the whole first hair cycle for a fetal human creates something called lanugo hair, which is not quite the same as the hair on our head, which is terminal hair. It's still unpigmented, but it's much longer and it's much thicker. And so here you can see an image of a 140-day post-conception fetus that has nice hairs all over its forehead. And some people may have had children who are born prematurely who are also covered in lanugo hair. And if you have seen that, then you would know that eventually this lanugo hair will shed, and then the hair that grows out of that same follicle in the next cycle is a much smaller vellus peach fuzz hair, as shown by my beautiful niece, Shoshana, on the right. And you can see in this blow up that engrailed is expressed pretty highly in the matrix in the bulb. This is a population of progenitor cells that will go on to differentiate and help to make the hair fiber in the inner root sheath. It's also very lowly in the bulge, which is a slowly cycling cell population, progenitor cell population. And so this is particularly interesting because given that engrailed seems to be upregulated at the time that eccrine placodes are specified, which happens to be just before the transition from lanugo hair to vellus hair during development, and the fact that we know humans have evolved a mechanism to express a lot of engrailed, sort of the obvious question is, well, what is the expression of engrailed highly in this existing hair follicle? What is that doing to the hair fiber and the hair cycle? So for this, we don't have any good systems to test this with human cells or human tissue. So we turned to a transgenic mouse line that Jana had also made when she was a postdoc. And so with the help of a graduate student, Reiko, and an amazing undergrad, Michiko, we have induced ectopic engrailed expression during the second hair cycle. So it's the first one after morphogenesis. So the hairs have fully formed in the back skin, and they are now undergoing just the normal cycling process. So using doxycycline, we're able to turn on engrailed only in the basal epidermis and follow the mice through to the next cycle. And here I'm just showing you an in situ for engrailed 1 mRNA in our transgenic. And for some reason it looks pink on my screen, hopefully not on yours. But there is no there's no baseline engrailed expression in mouse back skin. So there's nothing in the control and then a ton of engrailed in the transgenic. And hopefully what you can just tell by eye is that the engrailed and transgenic here on the right looks like its fur coat is a lot less dense. They look kind of scraggly compared to the littermate controls. So these mice were shaved when we started the doxycycline and we just let their fur coat grow in. So these guys, that's all it was able to do. But the hair follicle density is unchanged. So that's sort of like the difference between humans and chimps. But what we did find after much effort by Michiko to individually phenotype many hairs across many mice is that the hairs tend to be much thinner in terms of their width. And the way this is determined is in a mouse hair, unlike a human hair, they deposit these medullary cells across the shaft of the hair. And so you can bin them into widths of 1 through 5 cells across the shaft. And in red, you can see that the engrailed one transgenic is the engrailed one control. And they are also,
Heather,
they
you
tend to be
have
shorter.
1 minute.
Thank you. They tend to be shorter than the controls. And this is caused by a shortened anagen growth phase. So when we tracked them across the whole hair cycle, the transgenics were getting through their growth phase and exiting it much earlier than the controls were. So this has suggested to us that if in the mouse, engrailed one is not only required for eccrine specification, but it's able to reduce the size of the hair fiber even by some amount, then this could play a similar role in humans and could be involved in the transition from those larger lanugo hairs to vellus hairs around the time that engrailed gets upregulated in the skin more broadly. So today I've told you that both high eccrine gland density and a loss of fur are unique to humans, and they're required for evaporative cooling. And our work has suggested that these two traits are not only developmentally correlated, but they may have also co-evolved in the human lineage, and that engrailed one is very likely playing a role in both of these traits developmentally and evolutionarily, and could at least partially explain the coevolution of these two key phenotypes. And I will take questions. Thank you.
Thank you, Heather. That was a great talk. While I give the audience time to type in questions into the Q&A, I have one.
Mhm.
So I'm really intrigued by the structure of the eccrine glands. They have a long tube and a distal coil, and it very closely resembles the structure of adult salivary glands in Drosophila. What is the importance of the eccrine glands having that specific shape and structure? Do you know?
So the recognition that it's very similar to a salivary gland is apt. So it resembles salivary glands in mammals as well, and they're very closely related evolutionarily, and they start out very similarly developmentally. I suspect that having that coil attached to a long duct is just useful to be able to maximize like the surface area to volume ratio of the coil itself, and like kind of put it deeper in the dermis so that it's not sitting up high.
Yeah. Excellent answer. Thank you. All right. So we have a question from Chris Wright in the audience who asks, does Notch have human-specific genomic cis-regulatory regions?
That's a great question. I personally do not know the answer to that. I know where I could check, so I could get back to you. Possibly, but I would have to look. I'm sorry about that.
All right. We have another question from Veronica Sikora. I hope I'm saying that correctly. Did the engrailed one mice ever produce ectopic EGs on their back skin? And along a similar vein, I'm wondering if you've explored other models where EGs and hair cells are both present in the skin, and whether overexpressing engrailed in that case would decrease the EG-HF radius.
Great question. So no, we never ever see ectopic engrailed or ectopic eccrine glands in the back skin of engrailed one transgenics. That is because engrailed is required, but it is only sufficient when it has the right mesenchymal signals. And the back skin of a mouse just does not, the mesenchyme does not produce the right signals. We don't know what they are. And so that's actually my other project in the lab was trying to figure out exactly how the dermis is telling the epidermis to make an eccrine gland. We still don't really know, so stay tuned. So that's the first part of that question. And the second part, other models. Yeah, so in the mouse paw, so for most work in our lab, we use the mouse paw actually as a model, because in some strains of mice, they have 6 foot pads on the bottom, and those only make eccrine glands. But in between those foot pads, some strains of mice are able to make both eccrine glands and hair follicles. The problem there is that they actually form at around the same time. So it's not a good model for this particular question. And the question of whether overexpressing engrailed there can change the radius of the hair follicle inhibition field. We don't know exactly what the mechanism is, but basically what you get is a fate conversion from, instead of making a bunch of hair, if you overexpress engrailed in a strain that makes a lot of hair in the feet normally, they will actually be eccrine glands instead of hair. So it's a little bit tricky to test in the mouse. We are currently trying to get some more humanized or heterotypic recombinant systems up and running. They're just really tricky. There's organic typic rafts in skin are amazing, but they will not form appendages. So they're great for trying to understand just like basic stratification of the epithelium, but we can't form appendages with them. So there's not that many models for trying to test this, which is frustrating.
Yeah. Not that many models yet, right?
Yes.
Right. Sarah has a question in the panelist chat. How do non-sweating primates or mammals survive excessive body heat if sweating is so essential for human survival?
That's a great question. So lots of mammals have sort of evolved different thermoregulatory adaptations. So it sort of depends on the mammal, but you can think of like elephants have those huge ears that actually help them radiate heat. Rabbits also, like desert hares, also have very large ears that help radiate heat. There are some actually radiate heat out of like large horns that are highly vascularized and can dump heat that way. Primates tend to, especially apes, tend to use like behavioral mechanisms. So apes are generally very lazy and don't move around that much, like in the middle of the day. So they'll just find a shady spot and sploot on the ground and eat and wait until it gets cooler. So there's a lot of different ways that mammals have solved this problem. The thing that most people ask about is horses, because I think at least horse enthusiasts will know that horses are notorious for sweating, but they don't use eccrine sweating. They have more, there's another kind of skin gland called an apocrine gland, which is associated with the hair follicle, and it makes a different kind of sweat. And so they've actually evolved to be able to dump heat through apocrine sweating. So there's, many, many different ways that mammals have solved this problem.
That's really cool. I'm going to follow up on that and ask, what about aquatic mammals? And what about platypus? Do you know?
So platypus, as far as I know, they have sort of an apocrine gland-esque thing that's associated with the hair follicle, as well as their mammary glands also just sort of leak out onto the hair follicle. So they have multiple glands that are associated with the hair. For aquatic mammals, as far as I know, the bigger problem is staying warm rather than cooling off. If they need to cool off, like a large mammal, like a whale, will sort of like stick its fin up in the air to sort of radiate heat off of the fin. Yeah, but they don't tend to have a high density of like glands in the skin the way those of us on the land who need to like maintain like moisture and like good ceramides on our skin do.
Right, fascinating. I learned a lot today. If there aren't any more questions, I'd like to thank you, Heather and Austin, for your excellent talks. And I'd like everyone in the audience to know that this seminar has been recorded and will be available on the SDB website sometime next week. And I welcome you to please join us for next month's seminar on Friday, the 14th of November, when Olya Yarychivska from the Rockefeller University and Anthony Galenza from Stanford will present. Thank you all for coming, and I hope you have a good evening.